When you look at the United States, we use first generation cells that come from the postnatal donation. Can you speak to the benefits of that as opposed to culturing, which we don't do in the United States, that could then go down second, third, 15th generation? I'm just curious to hear your thoughts on that.
That's a great question. In our country, in our current regulatory environment, we are not allowed to expand or culture the stem cells, which initially, at first glance, sounds like a really fun and exciting thing to do. You can grow a stem cell farm. But the reality is, with every single generation of cell division and expansion, as exciting as it sounds to start with a few cells and end up with millions of cells, and having done this myself in a laboratory, I've been in a lab for the last 30 years of my life, probably, something like that. And it is true that you can put cells in culture, feed them the right things, and get them to grow, and end up with 1 billion cells when you started with 100,000. The reality, though, is that at the end of that expansion or cell culture, you have a very different cell, a very different cell population than you did at the very beginning. Those cells that were at the beginning were capable of doing that, but doing it in vitro instead of in vivo in our bodies, in our knee, our shoulder, wherever, whatever we're treating, is we still are not fully we do not fully understand what that final cell is going to be, and we can't aim it. And so by the time we harvest those expanded cells, they have a variety of characteristics that are very different from the initial cells that were put into the culture. One of those is how many divisions they have left. As simple as telomeres are, and as perhaps reductionist of the idea of telomeres is, our cells do in the genome. Every time we divide a cell, there has to be a copy made of that cell's genome or DNA. As that gets copied and copied and copied and copied, that actually reduces the number of future copies possible. That's one difference. The other differences are things that just start to fall apart. They just, enzymes and molecules and transfer RNA and so on, the cell is not able to actually continually maintain that perfect, pristine initial state. And until cell culture advances to the point where we can actually do that, those cells, I would prefer not to use them myself.